SD Emma Selner. She came to KCU with her BS in biology from Rhodes College in Memphis, Tennessee. While obtaining her degree, Emma performed computational chemistry research looking at novel inhibitors of aldehyde dehydrogenases under the guidance of Dr. Mauricio Cafiero. She was able to present her work both orally and via poster presentations at several conferences and meetings, including 2 ACS National Meetings. Emma was also chosen by the Committee on Undergraduate Research as one of 60 Undergraduate Research Advocates to petition for policy reform regarding undergraduate research funding during Posters on the Hill in Washington, D.C. Recently, her work was published in the Journal of Computational and Theoretical Chemistry titled The Effects of Ligand Charge, Orientation, and Size on the Binding of Potential Inhibitors of Aldehyde Dehydrogenase. Emma is an avid soccer player, having played College Varsity Soccer for 4 years. She was named first team NSCAA Scholar All-Region and was on the Southern Athletic Association Honor Roll. Since coming to KCU, she has volunteered her time at both the KC Care Clinic and Amethyst Place. We are happy to welcome her here today at the MKRC Journal Club platform for her to give her talk and present the paper that she recently had published. Thank you, Emma. I will turn it over to you and remind everyone, please mute your mics, and we will raise your hand with questions, and we will get those at the end. Thanks.
Thank you so much, Shelley. Appreciate that introduction. Yeah, everybody, welcome. Thank you so much for being here today. I know talking about computational chemistry isn't necessarily on everybody's to-do on a Friday afternoon, but it's something that really gets me excited, and I think it should get a lot of people excited. So hopefully I can kind of portray that today. So the title of my talk is Good News 2020, because I think we all need a little bit of that. Computer simulation furthers drug design in Parkinson's treatment. So as Shelley said, I am a second year on the Kansas City campus. And this work relates to the journal article that she talks about that my lab group worked on, as she said, at Rhodes College. I worked in a computational chemistry lab, basically looking at dopamine metabolism. And this work that I kind of passed on to my mentee, Caroline, then finished this and published this article. So it was in the November issue of the Computational and Theoretical Chemistry Journal. So I would just like to say a quick thank you. I know this is normally done at the end, but to them and to everyone that's here, just because it's really an exciting thing to talk about. So with no further ado, this is what we are kind of going to be talking about today. And let me just put my pointer on so you guys can see that a little bit better. So first, we're going to just talk about drug development in general. And I almost feel a little silly because I see so much of the pharmacology staff on here, but I think this will just really have a good basis for our mindset and my mindset when we were working through the project. So I'm going to talk about drug development, then move into Parkinson's disease, dopamine metabolism, which, as you will see, is greatly implicated in the pathophysiology of Parkinson's disease, and then talk about kind of the nuts and bolts of the paper, which is computational chemistry and computational modeling and ligand design, which basically means drug design. So hopefully if you're still awake, now we're going to move into drug development. So drug development in your stereotypical timeline, you have your drug discovery, where a compound is found. You move into your preclinicals, which is in vivo, in vitro, your clinicals, which is human trials, and if the drug does what it's supposed to do and it's not too toxic, it will be approved. So when you look at this from a number standpoint, it's really overwhelming. So normally you start with anywhere from 5 to 10,000 compounds, which that's a lot if you're trying to get to one that's going to work. And so, you start with those 5 to 10, and then only about 250 of those make it to preclinical, 5 of those make it to clinical, and about 1 gets through. So when you kind of look at the averages for what this timeline takes, it takes around 10 years and costs $2.6 billion, which hurts us all a little bit, I'm sure. But although 10 years might seem like a long time to us now, it's exceedingly fast compared to what drug development looked like in the past 50 years. So a really good example of this is L-DOPA. So L-DOPA is a drug that is widely accepted and used in Parkinson's treatment. And this is not really too much about the drug itself, but more so just its development and approval to kind of show you guys this process a little bit more. So it was originally discovered and synthesized in 1911, but it would not become commercially available until 87 years later in 1998. And so kind of looking at this, I think it really makes us appreciate the 10-year average that we have achieved. That being said, 10 years is still too long for a lot of individuals who are suffering from progressive and extremely debilitating diseases, for example, Parkinson's disease. So Parkinson's disease is an incredibly prevalent disease. There are 10 million individuals living worldwide with this disease. Today, 60,000 Americans are diagnosed with this every year. It is commonplace enough that I'm sure most of us know someone or a friend of a friend, a family member, someone who has been diagnosed with Parkinson's disease. So now that we're going to kind of get into the nitty gritty, this is a very simplified version of kind of physiology and the role it plays in Parkinson's. But the way that we thought about it when we were approaching our project was, okay, so you have this substantia nigra in your brain, and that makes dopamine. The dopamine helps the substantia nigra communicate with the basal ganglia. And the dopamine is pretty unique as a neurotransmitter because it can either be excitatory, inhibitory. Basically what it does is allows the substantia nigra to exhibit control over the basal ganglia, and that helps you control your motor movements. And like I said, this is obviously a really simplified version, but this was kind of the physiology that we looked at for our drug design. So when we look at Parkinson's disease pathology, we can see that the main culprit is that the substantia nigra is progressively degenerated. So without that part of our brain, we cannot be synthesizing that dopamine. And so when there is no dopamine, the basal ganglia loses that signaling. So either that stimulation or that inhibition that it needs. And this manifests physically as loss of motor control. And obviously there are a multitude of symptoms and manifestations of this disease due to its truly widespread neurological implications. But like I said, we're just going to continue to focus on the motor control losses for right now. So here are two, I think, really good examples of individuals with Parkinson's disease with some classic motor characteristics. So the first one on the left is showing an individual who has the classic Parkinsonian gait. So you can see her reduced step length, and I think probably the most obvious is when she turns, it's called defragmentation, where there's really short, shuffled steps, kind of a general postural instability and loss of control. There's a kind of hesitation. The other one that is also a classic sign is the hand tremors that an individual experiences. You can see here, this individual playing pool, riding, things like that become really hard when you do have these characteristic motor disturbances. So Parkinson's disease is extremely debilitating and greatly hinders an individual's quality of life and ability to complete basic daily tasks. So our focus then becomes, how do we help them? How do we fix this problem? And when we look at the pathophysiology, our focus should really be on dopamine and finding a way to increase its presence and therefore activity in the brain. So this is where L-DOPA comes in, and L-DOPA is the drug I mentioned earlier that's used to treat Parkinson's disease. So L-DOPA was designed to increase the amount of dopamine available to the brain to potentially restore and alleviate these symptoms to help these individuals regain a certain aspect of motor control. However, the world is not perfect. All of that dopamine doesn't go to the brain. And so when we add more dopamine, our body metabolizes more of that dopamine, inactivates it, and excretes more of it. So as you can just kind of see from this pathway, even though we are adding more dopamine, unfortunately, a really large amount of it is actually being metabolized and not even making it to the brain. And so dopamine metabolism truly decreases the amount of dopamine reaching the brain and decreases the efficiency of L-DOPA treatment. And so this kind of became, when we realized the next target, the next problem was dopamine metabolism. It wasn't getting the dopamine there that was the problem. It was the dopamine was getting broken down and couldn't do its job. So dopamine metabolism became our target for enhancing Parkinson's disease treatment. So just a little bit about dopamine metabolism. It's obviously a very long and complex pathway. So I'm just going to highlight a few specific steps that we really focused on for the main importance for dopamine metabolism and that offer potential targets for drug development. So here you can see, I know the wording's a little small, but right here it says L-DOPA. So this is the drug that is administered. And L-DOPA is metabolized by dopa decarboxylase into dopamine. And dopamine is what can cross the blood-brain barrier. So this is all good. If our goal is to increase the amount of dopamine available to the brain, we have the dopamine, but now we must look at how it is metabolized and broken down from there. So the further metabolism of dopamine really happens in two major pathways. So the first major pathway is the COMT pathway, which is this side of the image. And the other pathway is the MAO pathway, which is on this side of the image. And so with really extensive database searching, we found that arguably the most important enzyme that we wanted to focus on was ALDH1A1, which metabolizes dopamine. So it's basically the enzyme that's catalyzing this step from dopamine to this inactive compound, which then will be excreted. So as I said, ALDH1A1 became the focus of our project. So ALDH1A1 is, as I just stated, involved in the metabolism and deactivation of L-DOPA or dopamine. And the further we looked, we found that it was also associated with the conversion of all-trans-retinol to all-trans-retinoic acid. And that conversion has a lot of implications in different pathologies, in cancer, obesity, cataracts. The list was really too long to put on the slide, but it really emphasized that this enzyme is really important in the metabolism of a lot of different substrates, and for our purposes, in activating dopamine. So then we really truly knew the goal of our project was to inhibit ALDH1A1. So, in order to inhibit it, you have to understand the enzyme itself. So here you can see I have a little video. It's just literally a screen recording that I took of myself scrolling through the protein data bank. Awesome website, totally free. Go on. There are thousands of crystal structures of proteins that are available. So we really started there, and exploring this enzyme became the basis for our project. So computational chemistry and modeling. When I first started working on this project, and people asked me what I was doing, I really struggled to tell them sometimes. But this is kind of the most succinct definition that I think hopefully will really give you a solid basis for going into the rest of this. So computational chemistry is a branch of chemistry where a computer is used to simulate reactions to observe molecular properties. This is basically doing experiments on a computer. And this really speeds up the progress of the experiments and enhances our understanding of the enzyme itself when we can visualize it like I just showed you guys. And computational modeling is a mathematical model in computer science that allows precise simulation of complex systems. So this is basically the skin and the bones of computational chemistry. It is the math and the physics that are put into these programs that truly allow them to simulate and mimic an experiment to be as real as possible. And these models that we use are really the basis for the legitimacy of these programs and have been validated over and over for their accuracy. So as I said, the focus of our project is ALDH1A1, which is shown here. And so for our purposes, we're trying to inhibit this enzyme. So obviously you can see here, this is a huge enzyme. But really what we are trying to focus on is find the specific part of the enzyme that interacts with dopamine, so our active site. So as you can see here, I know that box is little, but I just highlighted a little part of the active site to show, even though this enzyme is what's doing it, it's a very specific part of this enzyme that's metabolizing dopamine. And so this is where our drug development comes in. So when we are trying to find a drug that fits into that space, we create these models of drugs like I have shown here. And this is the end result. And I'm kind of just trying to give you a very brief overview of what this looks like. So hang with me here, okay? So here you can see, this is our final project. So this is what I'm looking at when I was doing my experiments. So all of these kind of stick things you see, that is the ribbon structure from the active site in just amino acid well, it's a 2D here, but it was in a 3D form. So what we do, like I said, is we isolate the most important parts of the enzyme. So those are the amino acids here, kind of all these stick things. And then we go ahead and stick our drug in here right here. So you can see that's the drug is the ball and stick model, and the active site is more of just the stick. So that's just kind of a very brief overview of what that looks like. But most importantly is the implications that computational chemistry has on drug development. And as I have demonstrated, this field provides great potential for insight into enzyme drug or enzyme ligand interactions, and can be used to expedite drug discovery and hopefully safely decrease the amount of time between discovery and getting a drug to our patients that so desperately need it. So this becomes a question of how do we get there? Yes, we can, screen all these compounds, but we're still looking for that one. So how do we get there? How do we get to that one? And this idea is called the Pharmacoform Model. And this is basically the ensemble of common steric electronic features that are necessary to ensure optimal molecular interactions with specific biological targets and to trigger or block its biological response. So I just kind of like to think about this as a puzzle piece. We are basically trying to find out, think of the enzyme is the great big puzzle, and the ligand is the very last puzzle piece you have. You need to figure out, is it big? Is it small? What needs to go where? So you really have to figure out the ins and the outs of the enzyme to understand and make a drug that fits perfectly as a puzzle piece in that space. So this brings us to our ligand design and model development. So ligand design essentially means drug design. For those of you who don't know, you can think of ligand as a drug, a molecule, a compound. I might use some of these words interchangeably, but just understand that the ligand is the drug that we are designing, so don't get tripped up on that. So as I said earlier, we knew our goal was to design a drug that would competitively inhibit ALDH1A1. So again, this enzyme right here that is metabolizing dopamine into its inactive, to this inactive compound. And so we thought, well, ALDH1A1, we want to inhibit it. Why don't we just design a dopamine lookalike? We already know that it likes dopamine. So let's create something that's really similar to it. So that was the basis for our drug development. So as you can see here, this is a 2D render of dopamine. And so we said, you know what, let's keep this carbon group, this carbon ring, let's keep these hydroxyl groups, and let's change what's going on out here. So as you can see here, this is the LP series. This is the primary series that I worked with in my time at Rhodes in the lab. And so you can see here, we really kept that carbon ring and those hydroxyl groups. Did put this R group over here, and then this X just shows, even though there's only one compound shown here, that X means there can be an OH there can be an NO2 there, etc. So we really just kind of started with this basic idea of, okay, it likes dopamine, let's put something in there that looks and acts like dopamine with maybe a few changes to see if we can find something that ALDH1A1 has a higher affinity for than dopamine to allow that dopamine to be around for longer in the brain. So again, this is the image that you were looking at before, but I'm just kind of bringing it back again now, hopefully that we have a more full picture of kind of what doing this actually looks like and what it means. So again, here is our ALDH1A1 huge ribbon structure. So we isolate the active site, and next we have our molecule. So what you were looking at before is actually one of the drugs that we designed. So you can see, that carbon ring here, those hydroxyl groups here, and then those different R groups over here. So that's the idea, we have the enzyme, we know something it might potentially like, and so let's go ahead and see what it looks like. So again, you can see here the sticks are the different amino acids that were determined to be of the greatest importance in the metabolism of dopamine. So that means the one that kind of, pull it in, break it apart, that kind of idea. So again, the sticks are the ALDH1A1 active site, and the drug is kind of the bond stick model in here. So you can see, carbon circle, hydroxyl groups, etc. So basically what we do, again, this is a 2D rendering of a 3D model. So this is basically like we are simulating these experiments. I like to think of them in a little bin. And so there is fake water. As I said, all of those kind of theoretical models that are used that really take into account math and physics and all those things allow us to kind of put this in like a little air blob and simulate the interaction between the two. So this is kind of an example. Unfortunately, I do not have a video example of mine specifically, but I thought that this was a really good way to show a 3D interaction of the drug in the active site. So this is my favorite part. This is what I like to call the dance. And it's basically the interaction between the active site, which you can see here, they have the ribbons instead of the sticks. It's just a different representation of it. And then their drug is right here. So this is a video I just got off of YouTube that was actually done pretty early in the computational modeling days. But what the dance is you're going to freeze or lock the bodies of the molecules. So this includes the bodies of the active site and the drug. And this is just to ensure that certain parts of the molecules stay in one place. And this is so that it's not flying all over the place when we are doing these simulations. So you can really think of it as the core of the molecule being that trunk, the tree trunk that's rooted in the ground that stays in place. The second thing we do after we root that trunk of the molecule, we're going to free the side groups. So this is essentially like letting the branches blow in the wind. And this really allows for the enzyme and the drug to interact as realistically as possible. So it allows for, although they're staying in one space, they can move their arms, move their legs. They demonstrate when they are attracted to things, they move closer. If they are not attracted to things, they are repelled by them, for example. So hopefully this video will kind of give you a better understanding of what that modeling looks like. So you can see how the active site is not really moving along the sides here. And this part of the drug is also not really moving, but the ends are moving. As you'll see here, it's going to do a turn. So I'm going to play it again just so you can watch it again, because I think it's really helpful. But you can tell they're definitely a little restricted in their movement, but unfortunately that is one of the limits of simulating experiments. But this really shows how the interaction can really be demonstrated in real time for computational modeling. So this is a picture or a screenshot of the final pose of the dance. So we let these dances run for a certain amount of time, which basically means we give them plenty of time to get kind of into their perfect little spot. And so after simulating the experiment or running the experiment, which is where we basically just let the ligand and the active site interact in a water environment, we can see how the ligand or the drug sits most comfortably. And this positioning really demonstrates the drug's highest affinity for different molecules of the active site and its final conformation. So this idea was the basis for my project. So this is where it gets a little nitty gritty, but hang with me. So as you can see here, this is the drug. One of the drugs I've been talking about, carbon ring, 2 hydroxyl groups. So here specifically, we are looking at this R group right here and how it interacts with this amino acid, this is an amino acid that is a part of the ALDH1A1 active site. And so here you can see I kind of zoomed in. So this is the drug part. This is the active site part. And so I am basically calculating the distance between the drug molecule and the enzyme active site. And by doing this, I'm calculating interaction energies. And interaction energies are essentially the affinity of the active site for the molecule. For example, compared to when it started, we have our basis measurement, we run the simulation, it does its dance. And afterwards, when we look at these distances again, we can see, okay, Did it like the molecule? Did it pull it closer? Did it push it away? Did it flip it completely the other way? All of those things. And it gives us really incredible insight into the preference of the enzyme, and therefore we can really specifically target different parts of the enzyme for drug development. So this is, obviously when you're going to be starting a project, you need to kind of look at the basis for what is in the field nowadays. So the best competitive inhibitor to date for ALDH1A1 is an inhibitor called CMO26. So right here, and so you can see it in the ALDH1 active site. So again, the active site is the sticks, and the drug is the kind of ball and stick model in here. So we let CMO26 dance with the active site. And so you can see here we have our total interaction energy. IE stands for interaction energy. So we saw that it was negative 76. So what does that number even mean? So when you're looking at interaction energies, the more negative a number is, the more the enzyme wants that molecule in its active site. So the more negative a number is, the greater affinity it has for the molecule. So when we are looking at this, where it has a negative 76 for its interaction energy, a better or a stronger interaction would be negative 80, and a weaker or less strong interaction would be a negative 70. So this was kind of our benchmark for when we were looking at our drugs in the active sites. Okay, negative 76 is the line to beat. Can we do it? So this is an example of a LP molecule. So that's just the name of our drug line in the active site. So again, you can see it here. The A orientation, that just simply means which way the hydroxyl tails are facing. And that was really mostly a way for us to keep track of which way the molecule liked to sit. So, A was this direction and B was this direction. And so we really hit gold even on the first one. So this molecule right here, the LPCOH in the A orientation, had an interaction energy of negative 121. So that just absolutely blew CMO26 out of the water. But even though ones like that were really successful, I mean, ones with the same kind of base, so this right here, but with a different R group, had way less, had a lesser strength of affinity with the active site. And so even though, okay, so we can kind of maybe rule out this R group, or maybe we can target this specific amino acid in the active site, the cis302, but maybe it just doesn't really like that CN in that place. So even from right here, we can see, okay, the enzyme likes this molecule, specifically this amino acid really likes it, and it's really strong compared to the best inhibitor to date. So then when we started looking at just I remember one day I was sitting in the lab and I was thinking about it, and I was like, you know what? I know that this is 3D and all hypothetical, but our drug is small. It's a lot smaller than the CMO26, which is like I said, the best kind of inhibitor to date. And so I said, what the heck? Let's throw 2 of them in there and see what happens. And it did not disappoint. So here you can see.
Sorry to interrupt, but Dr. Kriener, I believe, has a question. Go ahead.
Oh, yeah. Sorry. Go ahead.
And yeah, I'm sorry to interrupt too, and I don't know if you mentioned this or not, but how long does each one of these computations take to fully run through the program for each drug or each molecule that you're talking about?
Yeah, no, that's a great question. It kind of depends. Obviously, the bigger, the longer it takes. We used to let them run overnight. Normally, they take around 6 hours, but it's the kind of thing where we would go into the lab in the morning, start the run, which is basically starting the experiment, and then log in the next day, and they would be ready to go. So it's definitely kind of a full day project, but it just kind of becomes this repetitive motion where, you submit one, and then you calculate the ones from the day before, and then so you can really keep it rolling, but it definitely probably, I would say, takes about 6 hours for the ones here that we were doing.
And is that on a fairly traditional computer, or were you using a more advanced computer based on modeling? Yeah,
so we were really just using kind of basic PCs. It was the modeling that we were using is based out of, it's a group called the Mercury group, and it's a computational chemistry modeling group. And basically we kind of have a base core computer that all of these different kind of, Rhodes is a pretty small school, so a bunch of schools went in and purchased this kind of like computer core that we all share. So the core actually isn't on site, but it is kind of the basis for running all of these simulations, but we can access them remotely from Tennessee, even though these are all in, I'm pretty sure it's Pittsburgh. So yeah.
Thank you.
Yeah, of course. And I'm so sorry with it being on my screen. I like can't see raised hands. So feel free to just interrupt me. I should have said that earlier, but so I'll just jump right back in. So as I said, I noticed one day, really small, let's try 2 of them. So this is the O orientation. So the O orientation basically means that they are opposite. And we actually saw some really interesting things starting to happen when we put them in opposite directions. So as you can see here with this one, the OH groups are on this side. So it's facing this way. If you think of my fingers as the OH groups, it's like this. And then the other one has its OH groups going the other way. So they're kind of sitting on top of each other like this. We started to see some really interesting ring-ring interactions between these 2 molecules that we think really contributed to this really, really high number of interaction energy that you're seeing here. And once we looked at this again, we really started to notice something about this phenylalanine 171 is very important in kind of the affinity and the metabolism of dopamine and of these potential inhibitors. So that is definitely something we identified as a focus for drug targeting moving forward. So just to kind of wrap things up, I hope everybody's still hanging in there. Hypothetically, if ALDH in the brain is inhibited by one of these novel ligands, which we have hypothetically proved it would be able to do by having such high interaction energies as a competitive inhibitor, the dopamine would be more slowly metabolized. And this could increase the effectiveness of L-DOPA therapy. And not only that, I think that this project and a lot of similar projects like this that are happening really shine a light on the abilities that computational chemistry has to really, like this says, harness the utility and powerful nature of rational drug design to approach drug discovery, really to make it a way, to cut down those 10 years, to cut down that $2 billion. To really make these processes faster and better, and at the end of the day, get faster results for our patients and their families, and hopefully make everybody's lives a little bit better. So with that, thank you. I would like to take any questions at this time. This is my thank you slide. All of my professors at Rhodes, our funding was based off of a National Science Foundation grant, and of course the Kansas City Research Department, all of their help, and my advisor, Dr. Dobson. So yeah, at this time, I'll take any questions. Thank you. Yes, please.
I have a couple of questions.
Yeah.
First of all, thank you very much for presenting your work. I thought you did a wonderful job making it very clear. And I think anyone could, I'm an example of this. I'm not really a structural chemist or biologist by any means, but you made it very easy to understand.
Well, thank you.
And a noob could come in and understand what you had to say. So thank you for that.
Yeah.
But getting to the kind of the idea of what it is that your data is showing, or maybe what it's going to lead to. What is the next step for this? So you've modeled this, on a computer. Where is this now going, or is it in the process of going?
Yeah, for sure. And now that you say that, I realize I forgot to put that future direction slide that Dr. Staudinger told me to put in. So sorry, but yeah. So basically, the idea behind this is that we have a partner lab. So we are the computational lab, and then upstairs there is a synthesis lab. And so our job is to basically slim down where we start from. I mean, that was just one example. I was one person in the lab. There were 8 of us. So, multiply all those drug compounds times whatever, it exponentially grows. So our job is to really narrow down what we think hypothetically will be the best. Just because, small school, money's tight, let's just make the most of our experiments. So what we do is we then, with our results, take them to them and say, okay, we think XYZ, for example, that LPCOOH, when we first saw that, we were like, okay, wow, this actually has a really high interaction energy. So we took that one to them and said, okay, this one is looking promising. They synthesize it and do some more like actual in vivo work. And then basically we kind of just publish it and wait for kind of a big pharmaceutical company to like maybe be interested, or just like honestly continue to find out more like about the enzyme. So that's kind of the next step. So like I said, it's just kind of building on all we have before. So those building blocks of kind of those really important amino acids of, maybe the best molecules. It really liked that COOH, R group, stuff like that. So just kind of passing those on to the next people in the lab and seeing where they can take it. So
awesome. So my other question was more of a global idea or global look at what it is you're doing. So when I looked at your enzyme ribbon structure, it looked to me like there maybe were 4 identical binding sites, just based on what it looked like to me, and I may be totally wrong. And then you've got your potential ligand and the modeling of interaction between the two. And this is my naivety on this field, as well as computers and what they can and can't do. But it seems to me that, you start talking about interactions between such a small portion of a ligand and a small portion of a molecule, enzyme, whatever. Are there ways to look at kind of your off-target effects before, it's probably expensive to synthesize an individual ligand and then get to a point where you're testing it in vitro or in vivo. So are there modeling processes that allow you to look at more off-target effects that maybe could even scale back the amount of drugs or potential drugs that go to the next step?
Yeah, I mean, I think that's a great point. Even though obviously it's kind of easy to talk about all of the pros, I think that is kind of a major con, just because it is so specific. Like you're kind of making all of these assumptions about where the ligand is going, exactly how it's binding, things like that. So I agree with you. I think that, that's definitely something that we need to look at. Like I said, by trying to base our molecules off of dopamine and that current inhibitor, the CMO26, we went a little bit based off of the safety profiles for those two to kind of determine, okay, even though that one that had a really high interaction energy had a bromine in there, okay, maybe we don't want to have a bromine in there because we don't know what else that's really going to get into. But, it's a computer, so let's throw it in there and see what the interaction is like. So I totally agree with kind of your hesitations about it. And I don't necessarily know of ways to look at it kind of on a bigger scale in regards to, just because I don't necessarily know how with computational things, the bigger you get, the longer it takes, things like that. So
right.
this
that was kind of connected to the question I asked about the computer size or whatever that you were using, whether or not that,
exactly.
I would imagine if you're going to throw more molecules into the gamish, you're going to start to need supercomputer type processing power.
So there's a lot of limits to it. But I think the points you bring up are really good, and obviously are concerns of ours and other people. You can run a simulation how many times on a computer, but what is it actually going to look like once it's in there, in the body, doing those sorts of things? So yes, I totally agree with you. There needs to be some progress made there as well, but I don't really have a good answer for other ways to look at it on necessarily a bigger scale other than hopefully someone with more money decides to try it out besides our small school.
So, student Dr. Selner, I have a quick comment and maybe an observation. So, thank you so much. That was an excellent seminar. I love your slides and the way you organized everything.
Thank you. Thank you.
Very clear, very clear.
You helped a lot. Thank you.
So, in my days at GlaxoSmithKline, this was a major initiative in silico modeling. And at that time, the idea was to make drugs more and more specific, no off-target effects. That was a paradigm a couple of decades ago that I think has shifted. And let's just make a brief observation about one or two drugs. For example, if acetaminophen or aspirin, either of them came to the FDA today, they would likely not be approved. And let me say this because some of the best drugs are dirty. They hit multiple organ systems at multiple levels. Dexamethasone might be another example, or phenobarbital, some of these sedatives. Very dirty drugs, some of the best drugs. That's just a comment. But what I know about rational drug design is it is a necessary first step that then enables those with all the money in the world, like pharmaceutical companies, to take it to the next level. And I see Dr. Schneider has his hand up. Thank you again, Dr. Schneider.
Emma, thank you for a great presentation. I'm sure I'm confident for you when you go for your looking for residency, and definitely that committee will have a member talk with you about your research. Remember, it always happens. So my question about, of course, that critical enzyme in metabolism of DOPA. So ADLH1A1. So question, I believe there were some studies, maybe some studies, which were looking at different single nucleotide polymorphism in this enzyme. Any of this SNP was associated with increased risk of developing Parkinson's disease. And why I'm asking it, because if so, maybe use that particular form of the enzyme, 3D, which is having SNP which predispose, may help you to design better inhibitor. That is my question.
No, I think that that's a great point. I'm actually trying to pull up something right now. Shoot, I don't know if I have the right thing for this, but yes, we did look at that. We did look at common SNPs or polymorphisms that are found in these receptors, both just in the general population and specifically in the population that has Parkinson's disease. And I'm so sorry, I can't find the data right now. I mean, this is what happens when, you graduate and get old and there's too many computers on or too many folders on your computer. But we did run some simulations with amino acid substitutions in the active site to see how greatly the interaction energy would differ with those polymorphisms, basically to ensure, hypothetically, if this drug were to make it all the way, it wouldn't be too hyperspecific or someone wouldn't need to be screened before taking it to ensure that it would have the same efficacy in those common polymorphisms. I think not only in specifically the individuals who have Parkinson's disease, but I think it's like 5 to 7% of individuals that were sampled had the same polymorphism in ALDH1A1, just randomly, totally healthy, all different ages. And so we looked at that as well. And from what I remember, I believe that the interaction energy was a little bit less, so less negative, as in it didn't bind as well, but it was still stronger than that other inhibitor. And I'm so sorry I can't find that poster on my computer right now. But it's a really great point to think about, especially when you're getting super, super specific with, what amino acid is interacting with it. That's definitely an important thing to keep kind of at the forefront moving forward. So I hope that answered your question.
Yes, thank you.
I thought it was a good question too, Dr. Schneider. And I think one thing, I don't know how many of you were here for the talk right before this, Dr. Joseph Miano, who was talking about mutations or polymorphisms or even single nucleotide differences in non-coding regions. And I think, as the field of drug discovery moves forward, it's going to be really important to think about within the enzyme itself. It may not be a different amino acid in the binding site, but maybe somewhere else within that enzyme that could alter the effect of the drug. And I know we have folks who look at those things and take that into account. But especially in the modeling, I think that's a great avenue because, as you say, you can just put it in the morning and the next day, look at it again and have your data out. It's a lot more efficient and probably less costly than trying to mutate an enzyme, using molecular techniques and creating a mouse with that mutation, blah, blah, blah. So just linking the two talks that we had together today.
No, appreciate it. Always welcome.
Any other questions or comments for student Dr. Selner? Carol Selner, I think it's time you unveil yourself and ask a...
I wonder if she left. She might have left. Yeah, everyone, my mom was on the call because I haven't seen her in a long time. So she was like, "Can I please tune in?" I was like, "Okay, just mute yourself, please."
You scared her away. You scared her away.
Yeah.
Anybody else? Last chance. 9 participants left in the room. Student Dr. Selner, congratulations on your publication.
Thank you.
I'll be sending you a link to your YouTube video as you move forward.
Okay, great.
Everybody with your speaking and scientific acumen.
Well, thank you. I really appreciate everyone being here. I know it's a Friday afternoon, so hopefully you can go get some fresh air if it stopped raining. So
I can think of nothing better to do on a Friday afternoon.
Well, I'm a geek, so I'm a fan, but teach their own. Okay.